Hi! In this video, we'll be talking about the Warburg Effect. How does it work in normal cells and cancer cells? We also provide a little bit of history, which it will include the person who came up with the Warburg Effect. I hope you can learn from this video, and thanks for watching.
The Warburg Effect in cancer. Learning material. Who was Otto Warburg? Normal cells, cancer cells. Warburg Effect. Who was Herbet Crabtree? Who was Otto Warburg? Otto Warburg was a German scientist who first described an unusual behavior in cancer cells in the 1920s. This unusual behavior is known today as the Warburg Effect. Let's first take a look at the normal cells in metabolism. Starting from glucose, it creates 2 ATP. Then move into pyruvate, and with the presence of oxygen, move into the mitochondria. Next to the Krebs cycle, creating 2 ATP, and then pass into the oxidative phosphorylation, creating 28 ATP. A process with a total of 32 ATP. Next, we have cancer cells in metabolism. Starting from glucose, creates 2 ATP. Then move into pyruvate. Here is the difference. Without the presence of oxygen, 85% of the cells will create lactate, and 5% with the presence of oxygen will move on into the mitochondria process. This will lead the cells to have 2 ATP made by glucose. Understanding the Warburg Effect. The Warburg Effect occurs due to the cell's mitochondrial malfunction. Therefore, cancer cells produce additional energy through increased oxygen-dependent glycolysis followed by lactic acid fermentation with secretion of lactate. In 1929, an English biochemist, Herbert Crabtree, extended Warburg's work and studied the heterogeneity of glycolysis in tumors. Crabtree concluded that in tumor cells, they did not only exhibit aerobic glycolysis, but there is also some fermentation, presumably due to environmental or genetic influence. Learn and be educated.